Hey everyone, Professor Davis here. We're going to be diving into the biochemical tests that we're going to be doing for lab 12 and 13. So the first set of tests I want to talk to you about are going to be the focus more for lab 12, and these are going to be rapid tests. They're going to be tests that we can do relatively quickly. They're going to take only a few seconds or a few minutes to get results, and so those are going to be the ones we're going to discuss first. So the first one I want to talk to you about is the catalase test. The catalase test is where bacteria can reduce oxygen to hydrogen peroxide. Now, when they reduce oxygen to hydrogen peroxide, that can become toxic to them. And so some bacteria do produce an enzyme called catalase, which can break down hydrogen peroxide into oxygen and water. So it's kind of like reversing it, making it not as toxic to them. Now, if they can do this, we're going to see that in the catalase test, we're going to see that catalase is present. That enzyme is present. So what we're going to do is we're going to add hydrogen peroxide to the bacteria. If catalase is present, it will convert the hydrogen peroxide to water and oxygen. You'll be able to see this by seeing that oxygen is produced through bubbles. Bubbles are going to be a positive test. So our procedure, we're going to take a slide. You're going to put a drop of hydrogen peroxide. You're going to then use your loop and you're going to pull off some of the bacteria, mix it in the hydrogen peroxide, and if bubbles form, it is positive. And so you can see here, there is going to be a positive and a negative. The positive has bubbles, the negative does not. So this is going to be one of the first tests that we're going to do, and we're going to see how many of the bacteria that are gram positive are going to also be catalase positive. The next test we're going to look at is the oxidase test. The oxidase test is going to be looking specifically for cytochrome oxidase, which is an enzyme that's found in that electron transport chain of some bacteria. Okay, so remember, bacteria do have an electron transport chain. It's just found in their plasma membrane instead of in a mitochondria because they do not have mitochondria. But this is going to be one of those electron acceptor proteins that could be present in the chain. So we're going to look to see if it's present. Now, the oxidase test does identify the bacteria that do contain the cytochrome oxidase. And what we're going to do is we're going to use an oxidase reagent. Now, this oxidase reagent is a really long term. Okay, it's a tetramethylene P phenylaldehyde dihydrochloride type of chemical. So it's a very large chemical name, but we're going to call it the oxidase reagent. Now, if we add this in, we see that it is what we call a redox indicator, meaning it's looking for that transfer of electrons that takes place. When oxidized, this forms the colored compound, what we call inosophenol blue. This indicates a positive test. So, if the blue is present, or that inosophenol blue is present, it's going to actually appear kind of purple on our filter paper. So how do we do this? What we're going to do is we're going to take some filter paper. You're going to take the oxidase reagent. It comes in a little capsule. You're going to break it apart because it comes in little crystals releasing the fluid. You're going to place some of the fluid on your filter paper, take your inoculating loop with some bacteria and rub it onto that reagent. Again, if it turns purple, then it is a positive test. If it does not, it's a negative test. One thing about this is you cannot lay down the filter paper directly onto your workspace. You need to make sure that you put it into one of those germicide containers because bacteria is going to be active on that. You just smeared it on there. So we don't want to spread that, so you can't lay it down on your workspace. Now, one thing to note is you have to read these reagents before they dry. You cannot let it dry completely and then get the results from this particular test. The last really fast test we're going to look at is called the coagulase test. The coagulase is going to be an enzyme that clots rabbit serum, okay, or rabbit's plasma. So we're going to use some rabbit's plasma. We're going to see if it clots it up. This enzyme converts fibrinogen, which is a soluble protein, to fibrin, which is insoluble, so you would see it clump up. This test is performed on staph species. So you're going to notice that we're going to have a couple of different staph species that may be present. And so you got to do the coagulase test. Now, this is the main test for us to tell the difference between Staphylococcus aureus and Staphylococcus epidermidis. Okay, Staphylococcus aureus is going to be positive for coagulase, whereas other types of Staphylococcus, specifically epidermidis, is going to be negative for this. All right, so the procedure on a slide, we're going to put a drop of water on one side and a drop of plasma on the other, and we're going to mix our colonies of bacteria into them. So we're going to mix it in there and we're going to look for clumps. And so you'll see that here we'll have a slide test and we'll look for that clumping type of factor. Okay, now sometimes we can't see the clumping really well, and so we would do a test tube test. If we do that, we see that we have to actually check those test tubes on a regular basis, like every half hour, hour, 2, 4, and even overnight to see if it clumps up. We should be able to see it pretty quickly on just the slides, but that's what we're going to be looking at for the coagulase test. So if you'll notice this top picture that you see here where it just looks cloudy, that's a negative test, whereas the bottom one where there's those clumps that are present is a positive test. So now let's look at the tests that are going to take a little bit longer. These are the tests that are going to be used for your gram-negative bacteria. And so you're going to have to inoculate them and come back and do the readings later. One of the first ones we want to look at is the McConkey's test. This is going to be an agar that is selective, meaning that it only allows gram-negative bacteria to grow on it. It inhibits gram-positive growth, so only your gram-negatives can grow on this plate. We also see that this agar is differential, meaning that it has a pH indicator. So as the gram-negative is growing, we're going to be able to see a color change take place. Bacteria that can ferment the lactose that's in these plates, that's a type of sugar, is going to appear pink on the plate, whereas the organisms that cannot are going to appear more clear. And so let's look at a picture of this. And so if you look here, you're going to notice that the gram-negative bacilli on the pink side are the lactose fermenters, so they have formed the pink colonies. On the other hand, the gram-negative bacilli are the non-lactose fermenters, and they form the clear colonies. All right, so again, these are gram-negative bacteria because it's selective, but differentially, you can see that there's a difference in how these guys will grow on the plate. The next test we want to look at are where we're going to see what else carbohydrate-wise that bacteria can ferment. So we're going to do a phenol red fermentation broth test, and this is used to identify acid and gas production from a variety of sugars. And the sugars we're going to look at are going to be things like dextrose, lactose, sucrose, and they could also put other sugars in there as well. So our procedure here is we're going to take the broths and we're going to inoculate them with a loop. So you have the broth, you're going to take your loop, sterilized loop, pick up some of the bacteria, and you're going to place it into the broth. You only need a few colonies to do this, but the tuber needs to remain up because there is a Durham tube inside. That Durham tube cannot tip because if you add bubbles into it, it skews your test, okay? So you got to keep this upright while you inoculate it. And then we're going to make sure we get all the bacteria off of our loop by kind of tapping the inner wall, rubbing it against it, making sure we get all the bacteria off our loop. We would then incubate this for 24 hours, okay, at 35 degrees Celsius, and we will stick it in just the normal incubator. So the results. How do we know if it actually fermented these types of sugars that were present? So these are determined by the color of the phenol red pH indicator. So if a sugar is fermented, acid is ultimately going to be produced. When acid is produced, it changes the phenol red to a yellow. So when an acid is produced due to fermenting the sugars, it's going to change from the red color to the yellow color. So if we take a look here, you're going to see that we have yellow versus red. Red is a negative test, whereas yellow is a positive test. You'll also notice in this particular case, you'll see the little tube on the inside. You'll see that it is higher and it has a bubble in it. This also shows that there is gas production. So we do see that it's twofold. We're going to see if they were able to ferment the sugars, and we're also going to see if they produce gas. So in this particular case, the tube on the left has been inoculated with E. coli, and so it gives you a positive test for the yellow and a positive test for gas production due to the bubble in the Durham tube. The tube on the right is inoculated, and it's just what it originally looks like with the red, and you'll see there's no bubbles in that tube either. So here's another example of a set of tubes. And so as you go through here, we can see that tube 1 is positive for acid because it's turned that yellowish. It's kind of orange, but it's a yellowish color. but you'll notice that the tube inside has no gas bubbles in it. So this would be positive for fermentation, or for acid, but it would be negative for gas. Number 2 is going to be negative for both acid and gas because it didn't turn enough. It's more of an orange color instead of yellow, so it did not turn enough. Now, given more time, it might, but we're not sure. Tube 3 is going to be positive for both acid and gas, so you can see that it turned a bright yellow as well as having the gas in the tube. And then tube 4 is uninoculated. It's not positive or negative. So you can see here that doesn't have anything in it. It's just that original red color. So this is the carbohydrate fermentation. The next one we're going to look at is called the IMVIC reaction. Now, when we look at this, guys, each of these letters means something different. So the I is going to be for indole. So we're going to test it for indole. M is for methyl red. V is the Vogt-Pascauer test. And C is the citrate test. Now, let's look at the first one where we're going to look at what we call MRVP. This is a media we're going to use where we're going to test the methyl red, so the MR, and we're going to test the VP, which is the Vogt-Pascauer. So we're going to test those first. So these tests are performed by inoculating a single tube. So you're going to have a single tube that says you have a single tube that says MRVP on it. You're going to take your inoculating loop, you're going to pick up some of the bacteria, and you're going to place it into the tube. Okay, mixing it up, placing it into the tube. This tube is then going to be placed in the incubator for anywhere between 3 to 5 days. So this one takes a while. This isn't one of those quick 24 hours. It's going to take a few days. So it's going to stay in there and it's going to allow the growth of the bacteria. We're then, after incubation, we're going to take that broth and we're going to divide it. We're going to take half of it using our pipette skills. Remember how you moved the water with the little pipette? You're going to pipette about half of the fluid out of one tube into another test tube. So there's half so that we can do both tests. We can do the MR test and then we can do the VP test. Okay, so we have to separate them. Once it's separated, you can do the methyl red or MR test first. With this particular test, this portion, you're going to use methyl red to determine if glucose can be converted into acidic products like lactate, acetate, or formate. So these are acids we're looking for. So again, the methyl red is a pH indicator and it will change from red to yellow. Okay, so we see that we're going to see that there's going to be a potential color change. Okay, so after methyl red is added to the tube, and a red color remains, we say it is positive for the MR test because methyl red is what we're looking for, so it's positive. This tells us that acid products were present in this particular test. Okay, so if acid is present here, it will remain red. Okay, if it changes to yellow, then the result is negative. Okay, so then the result is negative. And I'm going to show you a picture of these in just a minute. The next one I want to talk to you about is the VP test. VP is used to determine if glucose can be converted into acetone. This is going to be a more neutral reactant. Okay, so we're going to see if it's going to be positive for this. Now, with the VP test, you're going to use two reagents. You're going to use the Barrett's reagent A and B, and you have to make sure that you do them in order. You have to put A in first, then B. These are going to come in little capsules too that are going to be crystallized. You're going to have to break it to where it releases the liquid. Once the liquid is ready to be released, it's a little dropper. You'll drop some drops, the whole container of A in first, then you'll do the whole container of B. You do have to let this sit for about 15 minutes. After the 15-minute mark, if the acetone was produced by the bacteria, the top part of your fluid is going to turn a red color. So this is a positive test. If it is not, then it's still going to contain this normal color it normally will have. It's not going to have a color change at all. Because remember, the broth kind of has a yellow tint to it. It'll just remain that color. So in both cases, guys, for MR and VP, a red test is positive. Okay. So if we take a look here, you can see I have two sets. See how they halved the fluid in each. We ran the test, and so on this first side, we see that it is MR negative because it is yellow in color. But since it is red on the top of the VP test, this one is VP positive. On the other hand, if you look at the other type of bacteria, this one would be MR positive because it's red, but the VP is negative. All right, and so you're going to do both of these tests, looking at the MR for methyl red and the VP, seeing which ones are positive or negatives. But remember, the red color indicates that it is positive. The next test we're going to look at is the citrate medium test. Now, this one is one of my favorites in the sense of the color change. The colors on these are really bright and vivid and kind of cool, not just reds and yellows like we've seen so far, which are great and they're fine. It just gives a little different look. So when we're using the citrate media, these come in a slant, okay? So this is where the test tube's been dried or cooled at an angle, which means the agar is at a slant inside of the tube. So to inoculate this slant, you're going to use your loop, you're going to pick up a few colonies, and you're going to place it in there. You're going to drag it across the top in a zigzag method, inoculating that whole slant from the bottom all the way up in a zigzag pattern. You'll then place the cap back on loosely, and we are going to incubate this for 24 to 48 hours. Okay. After that time, it'll allow us to see if the color change occurs. So in the Simmons citrate test, the ability of the bacteria to convert citrate into oxaloacetate, which is a step of the Krebs cycle. So we're again keying on particular metabolism key for making ATP that these bacteria could do. We are going to look for the change. So if citrate is the only carbon source available to the bacteria, can it use it or can it not? If it cannot use citrate, then it will not grow on the media. But if the bacteria can use citrate, its sole carbon source that's in the media, then the bacteria will grow on the media and it will turn a bright blue. Okay, this is because the indicator is a blue color. Okay, it's going to look for that pH change. And so it starts out as kind of a greenish color. That's going to be our negative test, but positive is where it turns this really bright blue color. So again, this is where color coding your notes can be helpful because again, if you know the green is negative and the blue is positive. The next one we're going to look at is what we call the Myo or the Sim agar. We are going to use the Sim agar in our lab, but there are two. We see that the main difference is that they test this. The last test in both is a little different for a color change, but both tests can test for motility. That's what the M stands for in both of these. Motility is movement. The I in both tests is for indole. And then we see that for the Myo, we have an O. Okay. The O is going to stand for Ornuthen deamination test. Okay, that is the test we are actually not performing in our lab. Okay, but I am still going to talk about it, and I'm going to talk about what a positive and negative would look like. But that particular test is not one we're doing in the lab. We're going to do the SIM, where we're going to look at motility, indole, but we're also going to look at H2S gas creation. This is still going to do a color indication due to the H2S gas, but it's going to be different than what we saw in what we're going to see in the Myo test. Now the procedure, whichever test you're doing, you are going to use what we call a deep. This is where in the tube, the agar is going to be just filling it up. So it is going to be agar, it's not liquid in the tube. You're going to take your needle at this point, sterilized needle, pick up a few of the colonies, and you're going to do what we call a stab. You're going to go straight down to the bottom, okay, and then come straight back up. As straight as possible. That's what's important, and you have to be careful because, yeah, sometimes our needles are not completely straight, but do the best you can, okay? Put the lid back on loosely. We're going to incubate this for 24 to 48 hours. So let's look at some interpretation. Motility is the first thing you're going to be looking for. Has the bacteria, does it have the ability to move out and be motile in the actual agar? So you're going to hold the tube up to the light and look at your stab line to determine if there's movement away from that stab line. If it's non-motile, you will see the intact straight line down and back up that you saw. There'll be no movement from there. But if motility is present, you will see that there was going to be a kind of clouding of that original stab mark. There's going to be some movement out from there. It's spread out, and that's how you can use the light to help. You may also need one that's not inoculated for a comparison to see if it's cloudy or if it's still more of a clearer color. And so you can see the example here, okay? When we look here, the left tube is a result of a non-motile bacterium. Okay, so we see the left one has non-motile. You can see the stab mark that's present, whereas the right tube is going to be the result of a motile organism, where it's spread out. You can see that you can't even tell really where the stab line is. It's all spread out through the media. And again, seeing one that's not inoculated next to them would help you be able to distinguish these. The next test is the indole test. This one is going to be performed to determine if bacteria can break down the amino acid tryptophan into what we call indole. Now, after the myo or sim agar has been incubated for 48 hours, we are going to use a reagent called a Kovacs reagent. This is also going to come in a little tube. You'll have to break up the crystals, and you'll put a few drops into the media on top. Now, this is agar, so it's only doing the very top part of this. If bacteria can break down tryptophan to form indole, there'll be a reddish pink layer that will form at the top of the media. Now, failure to see this is going to be a negative test. And so if you'll notice, you'll see that the tube on the left is negative. It has no reddish pink line, whereas the tube on the right does. It is positive. And this is going to be a pretty quick test that you can see too. So you'll notice the results of reading these out are going to be pretty quick. The thing that makes these longer is they do have to be incubated before you can do the test. All right, so now this test is one we're not going to do. This is the part of the meo test we're not going to see, and this is where bromocresol, purple, is going to serve as our pH indicator, which allows for our detection of decarboxylase activity. So again, a particular enzyme because it ends in ase. Organisms that ferment dextrose can produce acids, thereby lowering the pH because of the acids being produced, and this causes the indicator to change from purple to yellow. Okay, so we do see there's a color change that can happen. So the presence of acids stimulates the decarboxylation of orthonine. We do see that it creates what we call alkaline conditions. So this will turn the deep from a yellow back to a grayish purple color. Organisms which do not produce decarboxylase retain a yellow or acidic color in their deep, meaning the deeper part of the stab. So here's an example. On number 1, we're going to check, because these are going to be tests, you can look at multiple things. So number 1's non-motile. Do you see how you can see the line straight down? The bacteria is growing in that line. You'll also see at the top it has a pinkish red ring. That means it's indole positive. We do see that we have carboxylation, and so it's still a yellowish color at the bottom, so this one is negative. Okay. On test tube 2, we see it's motile. Do you see how you can't see the stab line? It's kind of spread all the way through. This one does not have the pink or reddish layer at the top, so it is indole negative, but because the deep has turned a reddish kind of color, it is positive. Tube 3 is also going to be motile. It's spread through. You can't see the stab that's there. Indole is positive here because you can see the pink ring at the top. And then you see that decarboxylation of orthonine also took place because it is a reddish pink color in the deep. So a quick note here, the indole reaction is red at the very top of these tests, whereas the orthonine reaction is red at the bottom, 1/3 of the media. So you're going to do top versus bottom on these two types of tests. However, like I said, this is not one we're going to be doing in class. We are going to be doing the sim test. We are going to still do the indole. We're still going to do the motility in this particular test. So it will look similar to what we just saw. However, the color change is not going to be red and yellow here that we're going to see. We are going to use the hydrogen sulfide test, which is what the S stands for. This is with a sim agar. We can detect the production of that hydrogen sulfide gas, H2S. The agar contains peptone, sodium thiosulfate, and ferrous ammonium sulfate. And if hydrogen sulfide is produced, these are going to cause a black participant to form. And so you will notice that we have on the test tube on the left is negative. It's just the normal color that the test tube is going to stay. Whereas the test tube on the right is turned black, it is positive. So you're going to see a difference there. Now, if it's black throughout the whole thing, it's motile. Those particular bacteria have the ability to move. If it's just black in the stab, those are non-motile. Okay, they don't move. We would still do the indol test at the top of this with the Kovacs reagent in order to see if it is indol positive or negative. All right, one of the last tests we're going to do is called the TSI slant. This is a triple sugar iron auger or TSI auger. This detects carbohydrate fermentation along with gas and hydrogen sulfide production. So we're going to be testing a lot of stuff again, hopefully reinforcing what our other tests are showing. So to inoculate the TSI, you're going to use a needle. You're going to collect a little bit of bacteria and you're going to do a deep, but as you come out, because this is going to be on a slant, you're going to zigzag it across the top. So we do the deep down and then zigzag across the top. We will also incubate these with a loose cap for anywhere between 18 to 24 hours. So the slant does contain 3 sugars. That's why it's called a triple sugar or TSI. It has dextrose, lactose, and sucrose. It also has a pH indicator of phenol red in it. So when the carbohydrate fermentation occurs, if acids are produced, it's going to turn the phenol red yellow in this case. So if the deep remains red, so the deep part stays red, it indicates that no carbohydrate fermentation occurred and the bacteria is an obligate aerobe, meaning it has to have oxygen. And in this case, we term it as a KK, meaning it's alkaline in the deep and it's also alkaline on top. And I'll show you a picture of what the color means in a minute. If only dextrose is fermented, the deep turns yellow and the slant will remain red due to low acid production. And so we would mark this as a Ka. K is going to be the alkaline, A is the acid. Now, if lactose and or sucrose are fermented, both the deep and slant are going to be yellow. This is due to the amounts of acids that are being produced, and we mark this as an AA. Now, gas production can also be indicated in these. So if gas is formed during the carbohydrate fermentation, it will show in the deep either as bubbles or the auger will start to become cracked. And again, I'll show you a picture in just a second. H2S production can also be shown here, and this is going to be where the deep turns black. So you'll see the bottom is black, it's not red, it's going to have a black color to it. All right, so let's take a look at some positives, negatives, that kind of test. So over here, I have 4 different tubes. Okay, so if I'm looking at all 4 tubes, the first tube is going to be, you'll notice it's all red. So we mark this as a KK because it's alkaline. So alkaline is going to be the red. No sugars were fermented. You'll also notice that the auger is intact, so there's no gas. And because there's no black, there is no H2S produced. So that's a negative for all of them. Test tube 2, however, does have some dextrose fermented because we have the red color. So this is a Ka. And the reason we knew this was A before because it also turned black. There's also no gas production, but if it turned black at the bottom, like you see here, it is an H2S producer. Test tube 3 is a Ka. This one is going to be a dextrose fermentated. There's no gas produced again, but we do see that there's that red on the top, yellow on the bottom. So again, this is why it is Ka. K being the alkaline, A being the acid. Again, since there's no black present, that is going to be no H2S produced. Test tube 4 is the last one. You will notice that it is yellow through the whole thing. This one we mark as an AA. This means they've used dextrose, lactose, and or sucrose through this process. Gas was also produced. You see how there is a space at the bottom of this? There's not going to be the auger has been disrupted, it's cracked, it's pushed. That's showing you that gas was produced. And again, since there's no black present, this is going to be no H2S produced. So you're going to notice that on these tests, we're going to be able to read out a lot of different information. Now, this information is going to be useful because in the next two labs, we're going to take these tests and what tests positive and what tests negative based on what we do in lab, and we are going to create our own flow charts of these are positives, these are negatives, and what happens in each of the tests for each of the bacteria that we have. You then get to use those charts along with this handwritten part that you go through this PowerPoint with these tests on your final exam. This will allow you to know, well, if this test was positive, that tells me I've got this type of bacteria or these types. Then I do the next test to see what the next type is and so on, to where it narrows it down to one bacteria. It'll make a little more sense when I get to show you in person how it all works. But in the meantime, use this lecture in order to go through and fill in your notes that you get to use on that final. Okay, going through and seeing why we do the tests, how we do the tests, and what is a positive result versus what is a negative result for each test. Okay, now again, you can watch this video as many times as you want as you go through that. But if you do have any questions, please let me know.